Hello, so today we're going to be covering Chapter 5, Extensions of Mendelian Genetics, and what this chapter goes over are the exceptions to Mendel's rules. Are there exceptions? Yes, there are. It turns out that under certain conditions, dominant and recessive don't look like dominant recessive. So let's get going on that. So, Mendelian inheritance really deals with two inheritance patterns, the law of segregation and the law of independent assortment. Let's get my pen going here. Here's segregation, independent assortment. And you guys talked about that, or we talked about that in Chapter 3. It's also linked to Chapter 2. So the patterns and phenotypes that are going to be different than the ones that Mendel's laws predict. Remember independent assortment, back here on the last slide, right? Independent assortment and segregation are going to have to do with situations when, number one, the alleles don't have that simple dominant recessive relationship. In other words, one doesn't trump the other. Where the phenotype is influenced by the environment, say by like temperature or pH actually. Sometimes there's more than one gene that controls a single trait. A great example of that is eye color. We tend to think about like, oh, blue eyes, green eyes, brown eyes, but the reality is there's more than one gene that determines our eye color. If you just think about brown eyes, there are some brown eyes that are really dark brown, and then there's some that are kind of like an amber color. So we'll talk a little bit about multiple gene effects. Sometimes there are more than two alleles for a given gene that controls a trait. So think about Labrador retrievers. You have black labs, chocolate labs, and yellow labs. It's all fur color, but there are multiple alleles that give rise to the color of the fur. And also there are genes sometimes on the same chromosome. So it turns out if you look at a single chromosome, you might have a gene here and a gene here. What are the odds that they influence one another? An allele itself can be lethal. This is not a good thing. Lethal alleles do have a tendency to kill the offspring pretty early. However, some lethal alleles will cause death kind of in a protracted sense. So the offspring can survive for a while, maybe not long enough to pass on the trait. Sometimes though that they do live long enough to pass on the trait, to pass on the allele. The trait itself, it can be sex-influenced or sex-limited. Now, what is the difference? We're going to go into more detail there, but sex-influenced has to do with, say, for example, baldness in humans. The odds of becoming bald as a man are higher than the odds of becoming bald as a woman. However, you can get bald women. We're going to talk about how sex influences that trait. Sex-limited traits are traits that you should only see in either or. We're going to talk about plumage in birds for sex-limited traits. And then gene redundancy. Sometimes you just have more than one gene that does the same job. So we are going to start with a situation where you have mutant alleles. And the alleles are different from what we call wild type. Remember, wild type is the most common one that we see in the environment. Wild type may not necessarily be dominant, but oftentimes the wild type is the one that is more prevalent. A mutant allele can be recessive. If it's recessive, it'll tend to be rare in a naturally occurring population because oftentimes it causes what we call loss of function. Basically, the protein that the gene codes for is either folded wrong, it's inactive, it's broken, or you don't have enough of it, okay? So we're going to talk about later in the semester the regulation of how proteins are made from genes. How much do you get? And how are the cells sure that the right shape of the protein is made? It's really cool stuff. Now, we can also have mutant alleles that are dominant, all right? Dominant alleles, mutant alleles, can be a good thing because sometimes those alleles function better. We call that gain of function. Gain of function leads to evolution. So if, for example, a mutation happens in a gene and that mutation makes life better or easier on the organism that it's inside of, then that means that that organism has a gain of their functionality. The gene has given them a gain of function, which means they'll survive and reproduce better, which means that gene stays in the population. Some genes can antagonize a negative effect. So antagonize means be opposite of, work opposite of a negative effect. And then we have something called haploinsufficiency. This is a loss of function, but only in the heterozygote, all right? So big A, and little a work, but this one doesn't. It's not always clear why that's the case. Oftentimes, alleles that you find commonly or frequently, prevalently in the population, those are our wild type alleles, like I mentioned before. And you can have more than one of these. That's what leads to what we call genetic polymorphism. Poly for many, morphism for appearance, shape, right? So prevalent alleles usually make proteins at the right amounts. They work really well. Typically, prevalent alleles are gain-of-function mutations. And typically they're dominant, which brings us back here to gain-of-function. Isn't that cool? So let's look at, an example where you have a genetic disease that's caused by a gene mutation or an allele mutation. And so lots of genetic diseases tend to be recessive, okay? And this prevents that allele from making a fully functional protein. So let's say, for example, here's a chromosome, all right? And you have this allele A. This big A is going to make, so this is gene A. It's going to eventually make through several steps protein A, which has a specific shape, all right? And let's say that that's the functional shape. If, on the other hand, you have a situation where you have a mutation, where we're going to call that A prime, okay? It's similar, but there's just a mutation. Well, then protein A prime will end up, instead of looking kind of like this star shape, like that. The star and the squiggle don't do the same job. This means that you have a protein that does not function. It's non-functional. Non-functional in the traditional sense of protein A. All right, so let's look at simple dominant recessive relationships where the allele does not affect the phenotype of the heterozygote. All right, so big A, little a. All right. How can the wild type phenotype of the heterozygote be explained? So you're having this situation where you have big A, and little A doesn't work in this case. Big A is making enough protein for the cell to function properly. So that's really cool. That works. And that actually happens quite a bit. Little a doesn't necessarily always have to mean you have a different, it is a different outcome, but it doesn't mean exactly purple and white. Let's just put it that way. The heterozygote itself might produce less, more than 50% of the functional protein. The normal gene can be upregulated. So what are our two options? You don't need as much. You don't need 100% protein made. So in this situation, the heterozygote, all you need is one copy, you're good. Over here, in this situation, this is what makes situation number 2 different, all right, is that big A, let me erase that. I don't want to say it quite that way. Big A's gene activity or regulation or transcription, let's just call it transcription. We'll get to that later. Is increased. So that protein level is increased as well, all right, to a functional state. So this is a really cool thing because what that means is that the cell can detect that you only have one functional gene, so it's pulling on that, making it work harder. So here's an example: dominant functional allele, big P, which gives you purple, and the recessive defective allele, which gives you little p. Now, you would think that, little p, white's not really defective. We'll see later on in this lecture that really what's happening is there is an enzyme that converts protein number 1 to protein number 2, which is the purple pigment. And only big P makes this enzyme. Little p is mutant, and that enzyme's not fully functional. Therefore, you do not get the production of purple pigment. How cool is that, right? But we'll talk about that more in a little bit. Okay, so we're going to talk about penetrance now. So what is penetrance? We are going to be looking at large populations here. So when you talk about penetrance, you are looking at the percentage of the population that is displaying a phenotype. So the term indicates that the dominant allele doesn't always penetrate into 100% of the phenotypes of the individuals in the population. So an example of this, a measurement could be if you have 60% of heterozygous individuals, obviously carrying the dominant allele because they're heterozygous, right? Big A, little a. 60% of them exhibit the trait. Therefore, you have 60% penetrance of that given trait, of that dominant allele. Now, the deal is when you're measuring penetrance, okay, the measure, ooh, good grief. You know, I think one of these days I need to get a flat monitor here. Let me try. I'm actually flattening out my screen here so I can hopefully write a little better. So penetrance is measured using the heterozygotes population. And the good deal about that is that it's always going to tell you what that big A is doing. So most populations are what we call incompletely penetrant when you're looking at, not most, but many populations for many traits are incompletely penetrant for extreme traits. An example of this is polydactyly. Polydactyly is a situation where an individual has additional fingers and or toes. You will see this in humans. Oftentimes you see this, well, not oftentimes, but you do see this in cats. I've seen it once in rabbits. It's kind of cool. So polydactyly itself is an autosomal dominant trait. So it'd be like big P, little p, oops, that should be a P, or little p will show the trait. Affected individuals are going to actually have either full fingers all the way down to little tags, skin tags, for their 6th finger would be, and a lot of times that's the pinky finger. A single copy of this allele is usually sufficient to give an individual the trait. So you'll have 1, 2, 3, okay, and then a thumb. Oh, well, there you go. You guys, I'm a scientist, not an artist. What can I say? Actually, it should have been over here, right? Let me try to erase my additional 1st finger. Oh, that's not going to work. Sorry about that. In some cases, the individuals carry the dominant allele but don't exhibit the trait at all. Let's try this again. Here's a thumb, 1st, 2nd, 3rd, 4th pinky, and then another pinky, okay? Hey, how great would that be to play like bass? You have like a 6th finger, right? Cool. Here's an example of an individual with polydactyly. Here's the thumb, 1, 2, 3, 4, 5, and then, well, you can't see it actually because his other finger's in front. Actually, it should be right here, but this is a picture that's actually polydactylous. The reason they're showing you this is here is the extra pinky right here, all right? So the transmission of this trait started in the father's line. It was transmitted to this male and this female. This female passed it on to her offspring. This individual had to have been big P, little p, to pass the big P down to this individual, to this offspring, and this offspring. This represents that individual there, all right? So this is a pedigree diagram showing the phenotype. And yes, it's not the most perfect picture because, you know, here's the thumb, 1, 2, 3, 4, 5th finger right here. The pinky's kind of pointing out at you, but you can see it there if you look real closely. If you have a copy of the book, check out the book. So in this particular case, we're just showing you what the phenotype looks like in this individual. The pinky is much reduced compared to the original pinky, but these pinkies, the additional finger can be fully functional, or it can be, like I said, as little as a skin tag, or it can actually just look like a finger with no bone. All right, so that is penetrance. We are now going to talk about expressivity. The expressivity is the degree to which the trait is expressed. Going back to our polydactyly, right? Here is a small little pinky finger, all right? In the case of polydactyly, the person can have several extra digits, actually, depending on how highly the trait is expressed. We saw somebody with only a single extra digit, but you can have several. The individual can have several. And the person with the extra digit, with a single extra digit that is not fully functional would be considered low expressivity. An individual with 2 extra digits that are functional would be high expressivity. So expressivity is the degree to which the trait is completely expressed. Penetrance is the proportion of the population that show the trait. I can already tell you that on the exam, you should know the difference between expressivity and penetrance. So, what is the explanation for expressivity and incomplete penetrance? Well, it's not always really clear depending on the trait that you're talking about. In many cases, what ends up happening is that the phenotypes occur in a range, all right? We're always graphing out this normal curve. But the range is influenced by the environment and other genes. So depending on the trait, again, that we're talking about, you can end up with individuals or a curve that looks more like this, right? Or like this. It just depends on what the environment, what the influences are over that given gene. So now let's talk about the environment and what the environment can do to influence a phenotype. So environmental conditions that oftentimes we think about are going to be temperature, photoperiod, how much time the sun is out. A great example of this is fur color change, or coat color, in the Arctic fox. Arctic fox are a grayish brown color in the summer, but they're pure white in the winter. Arctic fox coat color, and this happens also with rabbits, Arctic rabbits. Their coat color changes so that they can blend into the environment. So let's first think about it from the prey's point of view, the rabbits. The rabbits need to hide from the fox, right? So the rabbits turn white in the winter to blend in with the snow. Well, that doesn't help the fox very much. And in fact, it wouldn't help the fox very much to remain brown or gray during the winter, because then the rabbit could see it coming, right? So the predator has also evolved a mechanism of changing their fur color seasonally to blend in with the background. So what you see here is environmental pressures crafting a coat color change based on temperature or the season. And it's basically an evolutionary arms race. Which organism is going to shift faster? Is the predator going to turn white faster so that it's stealthy when it's hunting rabbits? Or is the rabbit going to turn white faster so it can be stealthy going out into the environment to forage food and hopefully not get eaten by the fox, right? Cool stuff. Humans have a trait called phenylketonuria, PKU. And this is where you're unable to metabolize the amino acid phenylalanine. It turns out that phenylalanine is also called NutraSweet. Phenylalanine is an amino acid that tastes sweet. A system of PKU, phenylketonuria, is the development of what we call mental retardation or mental deficiencies that occur as children are growing, young individuals as they're growing, and they're eating regular proteins that contain phenylalanine. Since their body can't process phenylalanine, it'll build up in neurons of the brain, killing neurons, leading to individuals that have mental deficiencies. If it's detected early enough, individuals can be fed a diet low in phenylalanine. They eat a restricted diet, and basically they end up symptom-free. So those are two examples, coat color and phenylketonuria, for environmental impacts on the phenotype. Another sweet example here is temperature regulation on the number of facets or ommatidia in fly eyes. Now, yeah, how many of us are really that interested in how well flies see? I'm interested because, flies' vision helps guide them to where, to your food, frankly. It's also the taste receptors on their feet, different story. But basically, when Morgan was studying, well, after Morgan's initial research on the X chromosome, a lot of his students noticed that there were phenotypes in the number of facets or ommatidia on fly eyes. And so it's normal to see a phenotypic or number range under different environmental conditions. So the temperature that fly larvae, aka maggots, grow up at influence the number of ommatidia or facets on their eyes. So if not you, but if Drosophila grow at a lower temperature, 15 degrees, they end up with a whole lot of facets on their eyes. Now, what would a lower temperature reflect in the natural environment? Sunlight. So if these maggots, or not necessarily maggots, but eggs are incubated at a low temperature, say perhaps maybe in a dark area with very little sunlight, it would be advantageous for the flies to undergo metamorphosis, hatch out and then undergo metamorphosis, and develop a high number of ommatidia so that they can see better in the dark. On the other hand, organisms that develop at 30 degrees, double the temperature, right, might be growing in a more lighted environment. They may not need as many ommatidia, so they have fewer. Or at least that's the theory. So different temperatures affect ommatidial development in Drosophila eyes, and that's an environmental effect. Okay, folks, so that's kind of one of the ambiguous but kind of simpler examples. We're now going to move on to incomplete dominance, right? So incomplete dominance is a situation where you have a heterozygote, I'm sticking with green right now because I kind of like it, that exhibits the phenotype that's in between the two homozygotes. So big A, little a, little a. Let's say that this is pink. Oh, I'm sorry. Let's do something a little bit more extreme. Let's say that big A is red. They're going to be giving rise to red flowers. Little a is going to give rise to white flowers, right? Just like Mendel's example. So you would assume that big A, little a would give rise to red flowers, but in this case, it would be pink, right? So this actually happens in a plant called the 4 o'clock plant. The 4 o'clock plant shows incomplete dominance where the wild type allele, big R, gives you a red flower. The allele for the white flower color is W. So now we're looking at a different annotation. C for color, R for red, W for white. So when you cross a red flower with a white flower, you end up with pink flowers. And so instead of having what Mendel saw, right, Mendel looked at purple and white, and he ended up with big P, little p, big P, big P, big P. Your genetics teacher's losing it here. Big P, big P, big P, big P, little p, big P, little p, and then little p, little p. This would have been purple, 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 white, right? So he had a 3-to-1 phenotypic ratio. Over here, with incomplete dominance, you always have a 1-to-2-to-1 phenotypic ratio, right? Phenotypic. And so in this way, you can see that you have this intermediate range. And so intermediate dominance is characterized as a 1-to-2-to-1 phenotypic ratio when you cross two true breeding individuals in the P generation, red and white. Red and white give you pink. So 50% of the CR protein is not sufficient to give you a red phenotype. It gets diluted and looks pink. 1-to-2-to-1 phenotypic ratio is not the 3-to-1 ratio that we saw over here, like I mentioned. Therefore, we call that intermediate dominance. So whether a trait is dominant or incompletely dominant may depend on how closely the trait is examined. What does that mean? Well, you're looking at flower color, overall flower color. That's pretty superficial, actually. Let's look at pea shape instead of flower color now. Mendel actually looked at pea shapes in his original analysis. The guy was like ridiculous. He had a lot of time on his hands, I think, in the winter to look at phenotypes, and so he did, which is amazing. But what he visually concluded was that big R, and big R, little r genotypes gave you round peas, and only little r gave you wrinkled peas. Remember round and wrinkled peas? But let's look at the microscopic level. This is something that Mendel was unable to do at the time. So if you look microscopically at a round pea, what you're going to see is that all peas are not necessarily created equal. So when you look at what we're looking at here is the genotype. Here's our big R, little r, round versus wrinkled, right? Our phenotype is round and round for the dominant homozygous, and then round for the heterozygote, right? Wrinkled for the homozygous recessive. It turns out the reason why the homozygous recessive is wrinkled is because it contains a whole lot less starch than the round peas do. What happens is that inside of a seed, starch granules not only store energy for the emerging plant, but starch granules also hold water in the seed. When there's more water in the seed looks more round, just like homozygous dominant and the heterozygote. The homozygous recessive has so little starch deposited inside of the seed that it also buffers less water, which means the seed appears wrinkled. So in this particular case, you don't see a dominant recessive relationship. You actually see incomplete dominance, right? You see that there's not enough starch made. Over here, the big R, little r, the big R rescues the shape of this seed. So simple dominant recessive relationships and incomplete dominance can't always be picked out or analyzed at a superficial level. Sometimes you need to go microscopic or even look at the levels of protein content being produced as a consequence of the gene combinations, allele combinations inside of an organism. Okay, now we're going to move on to a situation called overdominance. Oops. So now we're going to move on to a situation called overdominance. My apologies, I pushed the space bar and it muted me, and so I could be heard for a second or two there. So overdominance is a situation where the heterozygote itself is actually better off than either homozygous situation. And so overdominance, you basically have big A, little a being the most favorable situation, right? So a great example of this is sickle cell anemia. So anemia is a situation where your red blood cells are not able to, well, you have a reduced number of functional red blood cells. Let's just put it that way. Sickle cell anemia, it's more complicated than that, but let's just focus on sickle cell anemia for now. It's an autosomal recessive disorder. And basically what it does is it produces an abnormally shaped hemoglobin. So there are two alleles for the hemoglobin A molecule. There's hemoglobin A, and then the mutation in this case is what we're going to call hemoglobin S, S for sickle cell. There's another hemoglobin B, that we're not going to focus on here. So what ends up happening is an individual that has HBS and HBS, okay, so the sickle shape, so that's both the mutation, right, S and S, produces all red blood cells that have a sickle shape. So a sickle shape is like a C shape. A sickle is like, it's what farmers used to use to cut grains. Sickle is what the Grim Reaper carries around at Halloween time, right? And what the sickle red blood cell shape does is it doesn't allow oxygen to hold on to the red blood cell very efficiently. And so what happens then is that the sickling phenomena shortens the lifespan of red blood cells. Red blood cells are considerably more fragile when they're in this shape here, in the sickled shape. The individual gets anemia, and the sickled red blood cells have a tendency to clump. And so they can block capillaries during circulation, resulting in strokes if it's really severe. And so what that does is it leads to a shorter lifespan in individuals. So here's what a normal red blood cell looks like. Nice donut shape there, except for the donut hole hasn't been totally popped out. And this is a sickled red blood cell. So in this case, when you look at the Punnett square here, you have two individuals, HbA and HbS, heterozygote, both mom and dad, and neither of them are going to be showing the effect of sickle cell anemia. Okay, they have reasonable amounts of hemoglobin. So in the four potential offspring combinations, you can end up with a homozygous dominant or a homozygous recessive. Okay, makes sense because you have two heterozygotes. In this case, you have an unaffected hemoglobin individual. This individual has sickle cell disease. Now, we're going to go to this malaria resistance here in a minute. In this situation, the heterozygous offspring are unaffected in terms of hemoglobin, so they can carry adequate amounts of oxygen, but they are also what we call malaria resistant. As it turns out, there's a little critter called a Plasmodium that looks like a worm, sort of, and the Plasmodium infects red blood cells, and so the Plasmodium they're able to easily infect red blood cells in malaria. And so when somebody gets malaria, red blood cells are destroyed, and the individuals develop a few different characteristics, but one of them is anemia. In the case of sickle cell anemia, the sickled red blood cell shape inhibits Plasmodium infection, so it confers malaria resistance. So over here, when the offspring is homozygous dominant, they don't have sickle cell anemia. However, they're also not resistant to malaria. Over here, these individuals, the homozygous recessive, they have sickle cell disease, but they are resistant to malaria, right? So they are malaria resistant. But because they have a tremendous number of sickled red blood cells, they also might have a higher incidence of stroke. So what nature has favored in these regions with high malaria is the heterozygote. The heterozygote demonstrates overdominance, right? Let's come back here, overdominance. Remember, you have a situation where the heterozygote has an advantage. They're more vigorous, I'm just going to cruise through here, than the homozygous dominant or the homozygous recessive. Even though the recessive is malaria resistant, like I said, they could still end up having a stroke. So it's more advantageous to be a heterozygote. You're unaffected, you still have some red blood cells. Some of them can get infected, but not all of them do, because the malaria Plasmodium can't spread, especially if it tries to infect a sickled red blood cell. So a sickle cell allele has been found in a real high frequency in Africa, where malaria is found. Well, the parasite undergoes that life cycle with a mosquito and other individuals, other organisms that have red blood cells. Actually, these mosquitoes can also bite dogs and other organisms. The red blood cells of the heterozygotes are likely to rupture when they're infected with the Plasmodium. And so it prevents propagation of the parasite effectively in a heterozygous individual. Pretty cool, huh? So you're better off if you're heterozygous, because these guys, while the sickle cell individual won't suffer from malaria, they will have sickle cell anemia, and the other individuals are not resistant to malaria. So at a molecular level, what are we looking at? Overdominance is due to the two alleles producing slightly different proteins. One is conferring disease resistance, in this particular case with the hemoglobin S. Other possible explanations for overdominance can be homodimer formation. Homodimer formation sometimes has to do with proteins that come together to do certain jobs. So this would be protein A, protein B, they're held together by protein C, and as long as you have all these things in place, the job gets done. You break one of them, okay? One allele. In fact, let's just focus on A and B right now. Let's get rid of C, if I can make it happen. There we go. If you just have A and B, if you break one, the structure falls. Doesn't function at all, okay? So homodimer formation can have something to do with overdominance. And then you can have variation in just the activity of one or the other. So let's first look at heterozygotes with one altered copy of the gene. So here's an example of an individual, right? A microorganism that'll infect a certain cell type that produces A1, all right? In this case, the pathogen can get in, just like sickle cell anemia and malaria, or it also happens with Tay-Sachs disease. Don't worry about that right now. It's just another example. The pathogen, in this case, when the A2 allele is present, the individual is resistant to infection. Therefore, the overdominant effect has to do with being heterozygous. Heterozygotes are resistant to tuberculosis in Tay-Sachs disease. Tay-Sachs is a pretty ugly disease, but you'll never get tuberculosis. So, and that, of course, is Tay-Sachs is elevated in areas where there's a high tuberculosis rate. Now, what if you have more than two alleles doing a job, making a protein that's important in a metabolic pathway? Lots of genes actually have multiple alleles, 3, 4, 5 different alleles. So one example, one really well-studied example, is the red blood cell blood typing situation, where red blood cells can be type A, type B, type AB, or type O. It turns out that the blood typing that we're talking about has to do with carbohydrates, okay, sugars that stud the surface of cells. And so we're going to call these carbohydrates antigens, okay? And so in this particular case, you have what's called antigen O, antigen A, and antigen B. I'm going to go back to that former slide in just a minute, okay? So let's go back to that former slide. AB, AB, and O blood types all have to do with the gene that codes for enzymes that make A, B, or the plain O carbohydrate tree, okay? So coming back to here, O is the basic tree, okay? N-acetyl-galactosamine, okay, is a sugar, galactosamine is a sugar, that gets added to the antigen here, to the O antigen, with the glycosyl transferase that puts A onto this, which gives you type A. Type B has its own enzyme that puts galactose, another sugar, onto antigen O. An individual that is type A, okay, is going to carry at least one of these genes right here. So we say that it'll be A. Each one of these lines represents a chromosome, or, so that's homozygous A, or this is heterozygous. We're putting a little I here to demonstrate that that one does not carry the A. That's just the way they do it in this field. Type, oops, sorry, my pen's not behaving. Oh, what did I do now? Oh, sorry, guys. Hold on. I will pause. Sorry, I figured out that I pushed a button that I wasn't supposed to. I have to write over here. Type B equals a chromosome with the B enzyme. So this is the B enzyme here. And you could be homozygous dominant, or homozygous, we would just say, or big B, little I. Now, type AB, so we're going to make this green and blue, okay, is that plus, you guessed it, this. And then type O, we're just going to give type O orange, right? Type O is just going to be little I, little I. They don't have an enzyme. They don't have the A enzyme or the B enzyme, okay? So this is the B enzyme. This is the A enzyme. There's neither the A or the B enzyme to put galactose or N-acetylgalactosamine onto the antigen O, so you don't end up with type A or type B blood. So in this case, you don't have a dominance effect, okay? There's no dominance here. A is not dominant over B. B is not dominant over A. You have a situation where only the little I is recessive because no enzyme is made. I is recessive to A and B, all right? But A and B are not dominant over each other, so we call that codominant, all right? So in cells that have both alleles, you'll have a mixture of phenotypes. Here is A and B. Antigen B is only present in B, and antigen A is only present in type A, and antigen O is the non-modified, all right? And so blood type O, non-modified. Blood type A is modified. Blood type B, modified B with galactose, all right? Blood type A is N-acetyl galactosamine. And then blood type AB has both of them on there. So how do you look at these dominant patterns among individuals with different parents? Can you figure out what your parents' blood type was? You might be able to. So if you look here at a female, the egg is going to contain an A blood type, but it's heterozygous, and dad is going to be type AB. The outcome of this situation, in this case, the sperm is going to only carry this or this, okay? In this example, it's carrying the B. So this individual will be type AB. This one's type A, all right? This one is type B, and then type O. So it's possible with heterozygous parents for offspring to represent all three blood types, all four blood types actually. So why is this important? Well, for safe blood transfusions to occur, the donor's blood must be appropriate to match the recipient. Type O, it turns out, can be received by everybody, okay, because type O does not react with proteins present in the recipient's blood. We don't need to go into all of that. On the other hand, type AB is a universal receiver, can take anybody. So O is the universal donor, okay? And then type AB is the universal receiver. A and B must have AB or O, all right? It is extremely life-threatening if you get the wrong blood type. So it's always good to know what your blood type is. If you don't know, you can ask your doctor. I do remember, this is just a fun little story. I mean, it's not actually fun, but I remember I had a student who had immigrated from Russia, and on her passport it said she was type B. And when we did blood typing in our physiology lab, it turned out she was type A. We ran 10 tests, type A every time. They wrote it down wrong on her passport. So had she gotten into a car accident, if they didn't check her before they did a transfusion, if that was necessary, they could have given her the wrong blood type, and that would have been absolutely life-threatening to her. It's a good thing she figured it out. Okay, so we're going to now transition to how sex, the phenotype of sex, affects traits. So it turns out that sex can influence certain traits depending on if you're male or female. We have sex-influenced traits and sex-limited traits. So let's start with sex-influenced traits. These are traits where the allele is dominant in one sex and recessive in the other. And so sex influence, in this case, is a phenomena of heterozygotes, all right? So if you are a man and you are homozygous recessive, then you're going to show it. If you're heterozygous, you may or may not. That's sex-influenced traits don't mean that they're sex-linked, which means these sex-influenced traits do not necessarily travel on the X or Y chromosome. In fact, most of them are autosomal. So the example that we use oftentimes are baldness patterns in humans. So it turns out that there's an allele that behaves dominantly in females, but is in males, but is recessive in females. And so gene type big B in males produces a bald male. The phenotype in females is, this table looks upside down. This should say non-bald. Ooh, that's a mistake. Yeah, And this should be bald. Wow. Good thing we're moving on to a new edition of the book, right? Hopefully they fixed that. So females who are big B are going to be non-bald, but males will be bald. Males that have big B, little b are bald. Females are non-bald. Little b, the males are non-bald, but the females can end up being bald because it's not often that you end up with a female this way. There is also effect of testosterone on this. Some females do become bald later on in life when they're heterozygous after menopause. And that has to do with the fact that as the female hormones are dropping, male hormones actually go up a bit, and that causes the effect of that big B on females. So it also depends on what time of life you're looking at the individuals. So pattern baldness appears to be related to androgens or the male sex hormone testosterone. Pattern baldness comes from the overexpression of the gene that makes testosterone, converts testosterone from its substrate into testosterone. And it turns out that testosterone can be highly poisonous to hair cells. Let's just put it that way. So this gene also binds to receptors on other cells. And so what that does is it produces baldness in individuals. Another situation in females is that sometimes on the adrenal glands, females can end up with a tumor, with cancer. And what will end up happening is it causes the adrenals to produce large amounts of androgen, male sex hormones, the precursor and testosterone. And what ends up happening is you'll have a female that is heterozygous, but she'll end up bald because of the upregulated male hormone. So when the tumor's removed, her hair will go back to normal condition. So females, if you catch it early enough, this is a recoverable situation. You can track the male pattern baldness or pattern baldness in families. So the pedigree, here's a pedigree for human pattern baldness, starting with a dad. And you can see very infrequently do you end up with a female that's demonstrating pattern baldness. So it's the autosomal nature of this that you can see in these types of pedigrees. We're going to talk about pedigree analysis more in another chapter. We'll get back to this type of thing later. Now, sex-limited traits. Some traits only occur in one of two sexes. So, for example, in males, in humans, breast development is normally limited to females, all right? We're not talking about the development of fat pads on the chest, on the pectoral muscles. We're talking about the development and the hypertrophy of mammary glands in females. Beard growth is normally limited to males. However, after menopause, some women do experience facial hair growth. And again, that has to do with an increased expression of testosterone in females. In birds, let's remove ourselves from humans now. I mean, we are talking about sexual dimorphism, the secondary sexual characteristics that are prevalent in males versus females. Let's look at birds. Males have typically more ornate plumage than females. So here's a hen and here's a rooster. They look nice, right? Ornate plumage. The males have a fluffier tail. They have a large comb on top of their head. They have this little gobble down here, right? Males are more brightly colored than females. One big idea is that this allows the male to be seen better, to be seen well by females. The plumage pattern in females allows them to blend in and sit and brood their eggs. So the effect of sex influences the appearance of these organisms. Having said that, there are rare situations where you have a hen-feathered male. Hen-feathered males exist in the population because they're able to more easily reproduce with females because they kind of blend in with the females. Roosters go after roosters. They will beat off other roosters so that other roosters can't breed with their harem, with their hens. If you have a male that can blend in with the hens because he looks like one of the ladies, well, then that male will be able to pass his genes on at least as effectively as this guy right here. Now let's talk a little bit about lethal alleles, all right? Some alleles are linked to genes that are absolutely essential for survival. If those proteins aren't present, then the organism just won't survive. About a third of our genes are essential for our survival. So if you think back to a biology class that you took and you think about the electron transport chain, if you lose one of those proteins, you are not making a whole lot of ATP, and that is essential for your survival. You will die. And so as an organism, maintaining electron transport is a big deal. So these are absolutely essential genes. Non-essential genes, well, they're not absolutely required for survival. You can have a mutation. I keep saying you, I mean the organism can have a mutation, but it won't kill the organism, all right? So a lethal allele is one that has the potential to cause the death of the organism, and those are going to be one of the essential genes, all right? These alleles result from mutation in essential genes. Essential genes are like essential nutrients. You need them for surviving, right? They're inherited in a recessive fashion. So some of these lethal alleles can prevent cell division. So that will kill an organism at an extremely early age, sometimes even before embryonic development is complete. Some lethal alleles cause an effect later in life. Huntington's disease does this. It's the progressive degeneration of neurons in the central nervous system. Central nervous system consists of the brain and the spinal cord, and what this leads to initially is dementia, and eventually it leads to shutting down spinal pathways that are important in, say, things like breathing. And so the earliest onset of Huntington's disease is usually around the age of 30. However, it's much more typically observed in 50 to 55-year-olds initially. Huntington's disease, as it progresses, is lethal, and it's lethal because it exerts an effect on neurons later in life. We also have conditional alleles that kill the organism when environmental conditions change. One of these is a temperature-sensitive condition. Now, we do have a little bit of this in humans, but our temperature has to go really high before we become temperature sensitive. That would be like heat stroke. In developing Drosophila, they can be killed, at most of them, at 30 degrees Celsius. However, and most of them survive if they're grown at 22 degrees. However, some genes allow them to grow at 30 degrees, as you saw earlier on with the Ommatidia experiment. Those conditional mutants thrive at higher temperatures because there's a mutation that allows their enzymes to not denature, not fall apart under the high temperature. So those are lethal alleles. Semi-lethal alleles kill some individuals, but not all of them. And basically what you have is a situation where environmental factors are going to either be preventing or inducing these semi-lethal effects. Lethal alleles, they don't tend to be Mendelian. An example of this is a Manx cat. A Manx cat does not have a full long tail. Some Manx cats have kind of a stumpy tail. Their tail isn't cut, actually. It's just a stump of a tail. And some true Manx cats have no tail at all. What this mutation does is it affects the development of the spine, and it affects the closure of the base of the spine as the organism is developing in utero. And so the mutation shortens the tail by closing up the spine early on. It's lethal in a homozygote. So if you have big M, little m in a male, big M, little m in a female, I should have put a picture of a Manx cat up. They're so cute. Big M, big M, big M, little m, big M, little m, little m, little m. These guys die because the spinal cord doesn't fuse during embryonic development, and that's not good for an organism. Oh, here you go. I guess I didn't have to write it out. So you have a 1-to-2 ratio of kittens that are born. These kittens you never see. You never see non-Manx, right? So big M also gives you early embryonic death, because in this particular case, I'm sorry, I was wrong. My bad. The non-Manx, little m, this was Manx is a dominant effect. Big M kills the individual. Heterozygous survives, and homozygous recessive is okay. This is a long tail or a normal tail, okay? I am sorry, I was thinking of another disease. And this is Manx and Manx. So one of the last things we're going to be covering are pleiotropic effects, the effects of multiple genes on a given phenotype. This is called pleiotropy, and a lot of our traits are pleiotropic. It's caused by one gene product affecting the function of other pathways. A gene's expressed, and it can either, that protein that's produced can either affect signaling inside of a cell. That gene product can affect different cell types, or the gene can be expressed at different stages of development. So pleiotropic effects, a great example of that is cystic fibrosis. Cystic fibrosis is a disease where the body doesn't produce, or the body produces really thick mucus, usually in the lungs. You also end up with very thick mucus in the digestive tract, reproductive tracts. And that all happens because there is an imbalance of chlorine or chloride ions, or chloride when they're Cl negative, of chloride ions being pumped out of cells, okay? So normally there is a protein, or there's a pump called a cystic fibrosis transmembrane conductance regulator, CFTR. This thing pumps, you'll have like a little box here, here's the CFTR. It'll take chlorine and it pretty much pumps chlorine out of the cell, okay? It transports them. Mutant cells do not transport chlorine effectively. So what that does is it causes the cells to produce thick, sticky mucus, also produces very salty sweat. So males will be sterile, females will also be sterile for other reasons. But in the case of males, there's real thick mucus that basically interferes with the proper development of the vas deferens, which is the pathway for transporting sperm out of the male reproductive tract. So a defect in this gene can have multiple effects. We're not going to go into all those details right now. We just wanted to use that as a quick example. Now, gene interactions occur when you have two or more genes that change the outcome of a trait. So that could be general traits like height, weight, pigmentation. You guys haven't really seen me, but I'm not even 5 feet tall, okay? I'm like a major shrimp. And so there are numerous of genes that have had an effect on my developing into my ultimate height or lack thereof. So multiple genes probably had something to do, plus environmental or potentially environmental effects might have had something to do with my developing my massive 4 foot 10 inches. So in terms of two gene combination patterns, epistasis, complementation, gene modification or modifying genes, and gene redundancy are all types of patterns that give rise to gene interactions. So we're going to look at these kind of individually, some of them in future lectures. So when you're crossing two genes, how do you get two distinct phenotypes? So one of the ways that scientists first started looking at this in the lab was going back to Mendel's peas, looking at the sweet pea. And Bateson and Punnett, yep, that's Punnett, the guy who came up with the Punnett squares, was looking at the purple flowers. They observed that true breeding varieties with white flowers always gave you what Mendel proposed, right? 3-to-1 ratio in the F2 population. These results weren't a surprise, right? Now, when you look at the F2 generation, when you are looking at something slightly different, a dihybrid cross, you're looking at a situation where you are looking at C and P, C for color, right? And P, wild type alleles. So you have a wild type allele, C and P, that are needed for the purple phenotype. Then you have mutant alleles, little c and little p. They wanted to know what these recessive alleles were doing in combination with the wild type to give rise to flower color. So that dihybrid cross was a little weird. They didn't get the 9-to-3 to 3-to-1 ratio that they expected. So what did they get? Instead of 9 purple flowers, they got 7. So what they figured out was that the big C, one purple color producing allele, is dominant to white, okay, to the little c, all right? The big P is dominant to little p, which also gives you white. So little c, little p masks big P, big C, producing a white color because these combinations knock these out. And so if the plant is homozygous for either recessive white allele, you're going to develop a white flower when you look at it with regards to a dihybrid cross. So epistasis in this situation gave rise to the following model. You have a colorless precursor molecule that interacts with enzyme C, big C. You end up with a colorless intermediate again. So this is protein 1, this is protein 2, neither of them that have a color. Enzyme big P is what results in the purple pigment. So what you have is an enzymatic pathway or a metabolic pathway. Remember, metabolism is the accumulation of all chemical transitions that occur inside of a cell. And so basically this metabolic pathway here demonstrated that two different enzymes resulting from two different alleles were necessary to give purple. It wasn't just purple or white. It was white, white, then purple. That was important here. So the recessive little c gives you an inactive enzyme. Therefore, you don't even get to number 2. And the recessive little p, if you have big C, so this would be here, in this case, the little c is knocking this out, so you don't have this intermediate number 2. If you have little p instead of big P here, you never transition from the intermediate to the purple pigment. And that should be, yeah, there you go. So if an individual is homozygous for either recessive allele, you end up with a white flower. So complementation is a situation where you have 2 mutant lines that show a phenotype caused by the mutation. So over here, the mutation knocks out either enzyme, kaboom, same phenotype, all right? And so what Mendel might have been working with was a situation like complementation. Gene redundancy is where you lose the function of an allele that may or may not have a phenotype. I mean, you know, one gene compensates for the other. We call these paralogs, or not homologous, but paralogous genes. And so homozygous knockout mice do not necessarily have a phenotype. So in this case, when you have a paralog, you have big A, little b, or big E, big B. You knock out A, you still have B to make up for it. Here, you knock out B, you have A to make up for it. You have to knock both of them out to see that you have redundancy occurring. We see this in Capsella bursa, which is the shepherd's purse plant. The gene is called shull, and what shull does is it changes seed shape. And so under this situation, only one variant, the homozygous recessive in both cases, are going to show you the mutant. And so this is a situation where neither gene knocked out gives you the effect. You have to have a double knockout to see it. All right, that's the end of chapter 5. I will be seeing you at chapter 6.